Hi everyone, welcome back. Today we are going to meet more neurotransmitters. In the last episode, we defined neurotransmitters and learned about them in general. Then we took a close look at acetylcholine, our old friend. Today we are going to meet new friends, including monoamines, amino acids, neuropeptides, and others. Then we are also going to look at diffuse modulatory systems. These are systems in which our new friends are very powerful. As in the first part of this episode, we require some preliminary knowledge of organic chemistry, especially functional groups and the representation of organic molecules. Here are some organic functional groups that are used for your reference. Let's first enter the family of monoamines. Monoamines are divided into three large groups, including catecholamines, histamines, and indolamines. The catecholamines have many neurotransmitters, namely dopamine, norepinephrine, and epinephrine. These are their chemical formulas. They are all called catecholamines because they all contain catechol groups, which is this group. The synthesis pathway is from tyrosine to dopa, then to dopamine, then to norepinephrine, then to epinephrine. DA is the abbreviation of dopamine, and NE is the abbreviation of norepinephrine. So first, tyrosine is like this. Tyrosine hydroxylase adds a hydroxyl group to tyrosine. This forms L-dihydroxyphenylalanine. We can see this because another name for tyrosine is hydroxyphenylalanine or L-hydroxyphenylalanine. Both are the same. Dopa is L-dihydroxyphenylalanine, meaning that there are two hydroxyl groups, which is very easily visible in this figure. Dopa is then transformed into dopamine via dopa decarboxylase. Dopa decarboxylase takes off the carboxyl group on dopa to form dopamine. This process requires vitamin B6 as a cofactor. Moreover, from this process, we can say that dopa is a precursor to DA. In a metabolic pathway, a precursor is a molecule that must be used in order to synthesize one molecule. That is to say, a precursor is the ancestor of a molecule. Dopa is a precursor to DA, therefore it has very desirable function. Just a brief mention here. In patients with Parkinson's disease, there's a deficiency of dopamine in the brain. However, we cannot directly deliver dopamine into the brain because the blood-brain barrier blocks dopamine. However, we can deliver dopa into the brain, and it can pass through the blood-brain barrier. Therefore, dopa is used for treatment of Parkinson's disease. Dopa is delivered into the blood, and it crosses the blood-brain barrier to enter the brain, where it is converted into dopamine. Therefore, we have made use of the fact that dopa is a precursor to dopamine. The next step is to use dopa beta hydroxylase to treat dopamine and make it become norepinephrine. Here, one hydroxyl group is added to the beta carbon. What is the beta carbon? Well, in amino acids, we first find the carboxyl group. The carbon on which the carboxyl group is attached is the alpha carbon. Then moving on, the next carbon is the beta carbon. And if there is yet another carbon, it is the gamma carbon. Here, although the carboxyl group has already been taken off, the OH is still added onto the beta carbon, so we call it beta hydroxylase. The final step is to make norepinephrine turn into epinephrine. Here, a methyl group is added. This is achieved through the enzyme phentolamine N-methyltransferase, or PNMT. In this process, there is some transport involved. Firstly, tyrosine is converted into dopa in the cytoplasm in the axon terminal. Dopa is converted into dopamine also in the cytoplasm. At this step, dopamine is transported into the vesicle. If the neuron is dopaminergic, then the neuron can directly release dopamine from these vesicles. However, if the neuron is norepinephrinergic, then dopamine is converted into norepinephrine within the vesicle. After that, norepinephrinergic neurons can release these norepinephrine from the vesicles. However, if the neuron is epinephrinergic, then we have to take norepinephrine out of the vesicle in order to synthesize epinephrine in the cytoplasm. A quick note, some of us may not be so familiar with the names norepinephrine and epinephrine. However, there is an equivalent way of saying these. Epinephrine is the same as adrenaline, and norepinephrine is the same as noradrenaline. Adrenaline is just what is synthesized by the adrenal glands above the kidneys. These are synthesized into the bloodstream when we are very excited. How are these catecholamines disposed of? Well, firstly, there is an extensive amount of selective uptake. Many of these are reuptaken into the presynaptic terminal. There are also two enzymes for degrading them. First, MAO, monoamine oxidase, in mitochondria. These are responsible for degrading these molecules after selective reuptake. We also have catechol-O-methyltransferase, or COMT, which is also responsible for degrading all three neurotransmitters. These neurotransmitters are distributed. First, dopamine. It is distributed mainly in the substantia nigra, ventral tegmental area, and hypothalamus. Norepinephrine is mainly distributed in the locus coeruleus and other places. Epinephrine in the medulla oblongata, sympathetic division of the ANS. Don't worry if you find these terms a little bit intimidating. We will talk about them later in an episode about neuroanatomy. Histamine. Histamine is the second large group of monoamines. It is synthesized from the amino acid histidine. Histidine is processed using histidine decarboxylase. The carboxyl group is taken off to get histamine. This process also requires vitamin B6 as a cofactor. The transportation is achieved using VMAT2, where MA stands for monoamine. This is basically vesicular monoamine transporter 2. Receptors are exclusively metabotropic. They're all GPCRs. The disposal is achieved through enzymes called DAO and HMMP. Finally, histamine is mainly functioning in immune responses, wakefulness and arousal, attention, learning, and other cognitive functions. It is most prominent in immune responses. And the third group of monoamines includes serotonin. This large group is called indolamines. Indolamines are synthesized from tryptophan, this amino acid here. Tryptophan is an essential amino acid. What does that mean? Well, essential amino acids are amino acids that cannot be synthesized by the human body. There are 9 essential amino acids out of the 20 that are used to make proteins. For example, phenylalanine is an essential amino acid. However, tyrosine is not, because tyrosine can be synthesized from phenylalanine by adding a hydroxyl group. Tryptophan is usually considered an essential amino acid. Now that we have seen many synthesizing pathways of neurotransmitters, why not try to deduce one ourselves? Let's try to deduce the synthesizing pathway of serotonin. Firstly, there are 2 steps in the synthesis of serotonin, also called 5-hydroxytryptamine. Please try to predict the structures of 5-HTP, 5-hydroxytryptophan, from the structure of tryptophan and the name of tryptophan hydroxylase. What is its structure like? There is a small tip. The number 5 here means that the hydroxy group is on carbon 5. The carbon number labeling is shown in this figure. Can you guess its structure? Well, here it is. This is a copy of tryptophan. Then we add a hydroxyl group to C5, the 5th carbon. This gives us this, which is 5-hydroxytryptophan. Now, let's try to predict the structure of 5-HT, also called 5-hydroxytryptamine or serotonin, from the structure of 5-HTP and the name of 5-HTP decarboxylase. Try to recall what decarboxylase did. What is its structure? Well, here is a copy of 5-HTP. The decarboxylase enzyme just gets rid of the carboxyl group. Therefore, erasing the hydroxyl group, we get this. This is our dear serotonin. The transportation of serotonin depends on VMAT2 as well, because it is also a monoamine, MA. Its receptors are mostly metabotropic. Disposal mainly includes serotonin transporter in reuptake. MAO, monoamine oxidase, can also be used to decompose serotonin after reuptake. As of function, serotonin is useful in the sleep-wake cycle and in mood and emotion. Therefore, serotonin is a very large target for antidepressant drugs, drugs that are used for treating depression. As we talked about before, the inhibition of serotonin reuptake using drugs called SSRIs can enhance emotion and mood and help alleviate or cure depression. Let's now meet some amino acids. Glutamate is synthesized from glucose. Glucose is put into a citric acid cycle, which is part of cellular respiration. It forms glutamine. Then, glutamine is converted into glutamate via the enzyme glutaminase. The transporter of glutamate is VGLUT, vesicular glutamate transporter. Glutamate has three main types of ionotropic receptors, namely AMPA, NMDA, and kainate receptors. These receptors are all named by their alternative agonists. AMPA, NMDA, and kinates are all agonists of these ion channels. There are also GPCR metabotropic receptors. They are called MGLURs, namely metabotropic glutamate receptors. The NMDA receptors of glutamate are especially notable. They have special properties. Usually, these ion channels are blocked by a magnesium ion. Even though glutamate binds to the channel, it does not open. The magnesium ion prevents the ions from flowing in and out. However, when there is enough depolarization, the magnesium ion is popped out, and now the cations can flow in and out of the channel. Moreover, this channel allows both sodium and calcium to flow in. The influx of calcium can cause diverse reactions in the postsynaptic cell. And NMDA is very important in synaptic plasticity, especially long-term potentiation and long-term depression. These are the long-term kinds of synaptic plasticity that we talked about in the last episode. Let's return to the properties of glutamate itself. Its disposal is reuptake by EAAT on glia and neurons. Yes, glia also has a part in recycling this glutamate molecules. It is converted back to glutamine using glutamine synthase. In terms of distribution and function, glutamate is widespread in the whole central nervous system, including the brain and the spinal cord. It is the most prevalent excitatory neurotransmitter. On the opposite side, we have an inhibitory neurotransmitter called GABA. GABA stands for gamma-aminobutyric acid. This is its one-step synthesis pathway. Let's try to decompose its very long name. Acid means that there is a carboxyl group at the end. Butyric means that there are 4 carbon atoms in this molecule. These are the 4 carbon atoms. Amino means that there is an amine group as well. And finally, gamma, as we have mentioned earlier, is the numbering of the atoms. The carbon atom closest to the carboxyl group is called alpha. This is the alpha carbon. The second closest is the beta carbon, and the third closest is the gamma carbon. The amino group is attached to the gamma carbon, ergo the name gamma-aminobutyric acid. By the way, all the 20 amino acids that are used for the synthesis of proteins in our bodies are alpha amino acids, meaning that the amino group is always attached to the alpha carbon. Here, GABA is an gamma amino acid, therefore it is different from those alpha amino acids. GABA is received by receptors such as GABA A, GABA B, etc. GABA A receptors are mostly postsynaptic, while GABA B receptors are mostly presynaptic. GABA B receptors allow a response to the neurotransmitters that the neuron itself has released. The most notable one is GABA A receptor, chlorine channels. These chlorine ion channels have agonists such as ethanol or alcohol, benzodiazepine, barbiturate, and neurosteroids. These all have some pharmaceutical implications. It also has an antagonist called strychnine. In terms of transporter, the transporter's name is VGAT. GABA is disposed by GABA transaminase. It is widespread in the whole central nervous system, just like glutamate. It is the most prevalent inhibitory neurotransmitter in the central nervous system. However, in the peripheral nervous system, many times glycine is used as an inhibitory neurotransmitter. Glycine is the simplest amino acid. It only has an amino group and an carboxyl group, and it is very simple. That is the most used inhibitory neurotransmitter in the peripheral system, so be sure not to confound GABA and glycine. Question time. It's time for a little recap to all the new friends that we've just met. Now, which of the following statements are correct? A. GABA is synthesized from glutamate, so it is an excitatory neurotransmitter. B. Epinephrine is a precursor of norepinephrine. C. Part of the synthesis process of norepinephrine takes place in the vesicle rather than in the cytoplasm. D. Glutamate only has ionotropic receptors. Which ones are correct? The correct one is C. For A, GABA is synthesized from glutamate. That is true, but GABA is an inhibitory neurotransmitter, while glutamate is excitatory. B. Epinephrine is not a precursor of norepinephrine. On the contrary, norepinephrine is a precursor of epinephrine. C. Yes, part of the synthesis process of norepinephrine takes place in the vesicle. The specific step is the synthesis of norepinephrine from dopamine using dopamine beta hydroxylase. And D. Glutamate has both ionotropic receptors and metabotropic receptors. The metabotropic receptors are called MGLURs, metabotropic glutamate receptors. Finally, we get to another family of neurotransmitters, namely neuropeptides. Neuropeptides are usually synthesized in the soma, just like every other protein. It is transported using anterograde transport with the protein kinesin. Its receptors are usually metabotropic, and its disposal is usually enzyme degradation. An example are opioid receptors for endorphins. There are delta, mu, and kappa opioid receptors, which are GI-coupled GPCRs. And finally, let's take a look at something different. We will look at two special kinds of neurotransmitters. Firstly, endocannabinoids. Endocannabinoids are small molecule neurotransmitters. However, they have the special property that they are very lipid soluble. Therefore, they can pass through the cytoplasmic membrane and diffuse to other neurons. Their hydrophobicity facilitates this action. More specifically, they can diffuse back from the postsynaptic neuron to the presynaptic neuron. This is a process called retrograde neurotransmission. It allows the postsynaptic neuron to tell the presynaptic neuron how it is feeling. This is like a rating process. The presynaptic neuron feeds the postsynaptic neuron with neurotransmitters, and the postsynaptic neuron is now rating the presynaptic neuron, giving it feedback. We also have nitrogen monoxide. It is also a very, very small molecule and ergo can diffuse across the neuronal membrane. It is synthesized from arginine by nitrogen monoxide synthasase. Arginine is a nitrogen-containing amino acid. The mechanism of the function of NO is to activate soluble guanylyl cyclase. This then increases the amount of cGMP, because cGMP is now synthesized from GTP. cGMP in turn acts as a secondary messenger and increases the function of protein kinase G, which phosphorylates other proteins, like other protein kinases. Nitrogen monoxide, NO, is a free radical. It has unpaired electrons, therefore it is very reactive and can react with other proteins and other components of the cell. Therefore, it can be harmful in large quantities and are rapidly degraded after they're used. One of the key functions of nitrogen monoxide in the body is vasodilation, especially in erectile tissue. It helps vasodilation and the hardening of those tissues. Finally, we can see a place where neurotransmitters are applied in an extensive amount. Those are diffuse modulatory systems. In these systems, one neuron can make synapses with many neurons, up to the number of thousands, 10,000s, or even hundreds of thousands. This is a very good example of divergence, in which one neuron diverges into many neurons. In contrast, in the future, we will see examples of convergence, in which many neurons converge to make synapses on one neuron. Divergence helps diffuse modulation. A few neurons can modulate the behavior of many neurons in the brain. There are diffuse modulatory systems of acetylcholine, serotonin, dopamine, and norepinephrine. Let's take a brief look at them one by one. Acetylcholine. There are cholinergic neurons in some areas, such as the medial septal nuclei, the basal nucleus of Meynert, and the pontomesencephalo tegmental complex. These complexes project to nearly the entire cerebrum in the entire brain and modulates the excitability of the cortex. It also helps with arousal and learning and memory. Serotonin mainly exists in the RAF nuclei in the brainstem. It mainly modulates mood and emotions. Dopamine exists in two places, the substantia nigra and the ventral tegmental area. In the substantia nigra, it projects to the basal ganglia. This is part of the motor system. A deficiency of dopamine in the substantia nigra is correlated to Parkinson's disease. In the VTA, ventral tegmental area, there is a projection to the forebrain. This is called the mesocorticolimbic system. The system is in charge of reward. For example, if you eat something and find it to be very delicious, then you will eat it again. In this process, your reward system is functioning, because something is good has triggered a reward sensation, and you are likely to repeat this action to gain reward again. Finally, norepinephrine. It mainly exists in the locus coeruleus, which is the Latin for the blue spot. It is a very small cluster of neurons in the brainstem. This diffuse modulatory system is responsible for attention, arousal, sleep-wake cycles, learning and memory, and many other functions. Now, a final recap question for this episode. Which of the following statements are correct? A. Diffuse modulatory systems are very good examples of divergence. B. NO does not exactly fit into the three-condition definition of neurotransmitters. C. Neuropeptides usually have ionotropic receptors. D. Retrograde neurotransmission does not involve exocytosis of neurotransmitters. Please take a moment to pause and think about it. The answer is A, B, and D. A. Diffuse modulatory systems are very good examples of divergence indeed, because one neuron can connect to many neurons. B. NO does not exactly fit into the three-condition definition of neurotransmitters. In the first place, it is not emitted by the presynaptic neuron. It can even be emitted by the postsynaptic neuron, which is something weird. That is why we define it to be a special neurotransmitter. C. Neuropeptides usually have metabotropic receptors instead of ionotropic ones. D. Retrograde neurotransmission does not involve exocytosis of neurotransmitters, because these neurotransmitters involved, like endocannabinoid or NO, are lipid soluble and they can diffuse across the membrane. In this episode, we have met more neurotransmitters, including serotonin, histamine, dopamine, norepinephrine, epinephrine, glutamate, GABA, neuropeptides, endocannabinoids, and nitrogen monoxide. We have also seen the function and structure of diffuse modulatory systems. Coming up in the course, we're about to dive into the world of the functions of the nervous system, including sensory, perception, and motor functions. However, before that, we need to first have a way to navigate the nervous system. So in the next episode, we will have an overview of the neuroanatomy, and in the episode after that, we will take a look at the development of the nervous system and how neuroanatomy is formed. Then we can truly enter the world of sensory, perception, and motor functions. See you again in the episode of Neuroanatomy Overview. Bye!